Buongiorno a tutti. Allora, intanto volevo ringraziare la SIE per avermi invitato a presentare i miei risultati, o comunque alcuni dei miei lavori, davanti agli esperti nazionali di comportamento animale, che è un motivo di orgoglio, soprattutto per un italiano. E farò la presentazione in inglese e racconterò un pochettino di alcuni risultati riguardo le cause e le conseguenze dell'individualità comportamentale, cercando di metterle un pochettino in parallelo a quella che è stata l'evoluzione del nostro campo di ricerca negli ultimi anni. But so before I start to talk about actual science, I just wanted to say two things about me. So my name is Giovanni. I was born in Rome. I'm a father of two, soon to be three. And as Cristiano mentioned already, I am based in Australia at the moment. I just moved recently in Melbourne, but I spent the past 4 years on the opposite side of the continent, on the western coast of Australia, in Perth, where I met Clelia a few years ago. And at the moment, I'm lucky enough that I collaborate with colleagues from 2 different institutions. So I'm a research fellow at both Monash and at Tusha. And as I mentioned, I'm an adjunct to University of Western Australia, where I've been in the past, but got a PhD in Germany. Spent a few years in New York working on robots and how to use robots to study the behavior of live animals. Made just a little bit of a break in Spain to study something there, but the truth is that the main universities that I've attended, the main classes, have been in Italy, where I got like my bachelor's and my master's. So Cristiano before was saying, you know, a long time spent abroad. And in fact, my mom every day keeps asking me why I choose this career path that keeps me away from home for so long. But I think if you look back in time, the answer is kind of obvious. The reason is that I loved animals since I can remember. And so despite I've been living in the center of Rome, my house has been a zoo since I was a child, collecting and keeping all of the possible animals. I don't have a picture of my goat, but I had a goat when I was like 6 or 7, I assume, in an apartment. It was kind of funny. And yeah, so I knew that I wanted to do something with animals, not necessarily, you know, a scientist, but definitely something that I had to do with animals. And I think that this man, Professor Alleva, has been very influential in the choice I took to go for an academic path. So when I was in the middle of my bachelor at Sapienza University, he was actually studying his very first course of animal behavior that didn't exist before. So I was lucky, I chose it. And so I had a chance to see behind the scenes what is the scientific framework that is behind, you know, observing just an animal and the behavior of this animal. And so I joined this group, and in the experimental thesis of my bachelor and my master, I studied stereotypic behaviors in captive parrots. And believe it or not, all of these parrots were actually mine. So this is a picture of my balcony. And for the happiness of my neighborhood, you know, I've been there observing like how changing the space available and the social opportunities of these parrots, we would see like, you know, variety in their stress responses. But this also actually gave me a lens to see through what's behind the observation, what is the data collection, the statistical analysis, and also the communication of the research to the scientific committee and the community of scientists that work on a similar topic. Potentially hoping to find somebody as excited as I was to receive this information, as I tried during my dissertation, hoping that my committee actually was as excited as I was. From the picture, Enrico doesn't seem as much, but I know that he enjoyed it. But anyway, today I moved a little bit far away from parrots' behavior, and the main research that I do is focused on understanding how animals adjust to changes in their surrounding environment, with the hope of getting some insights to predict how group of animals, populations, or even entire species might respond to changes in the environment that is happening very quickly nowadays. But the main thing that I'm interested in is how individuals differ from one another and how do they respond differently from one another in these potential changes. And through these few examples I will show you now, I will try to explain why I think this variability is important, variability within a group, or within a population. The reason is that this diversity in behavior that we find within animal groups is important for resilience in terms of changes, but also the difference in how individuals can adjust, can cope with changes, gives them different strength to actually coping with an actual change. And the main models I am using in my research now are fishes, for many reasons. And they have been very useful to test how environment alters these behavioral phenotypes. In some of my research, I had a chance to capitalize on the use of biologically inspired robots as a stimuli to then integrate this virtual or this robotic component into a live system and then observe the reactions. So when I started my PhD in Germany, I was quickly introduced to the field of animal personality or behavioral individuality. And it took me a little bit to understand what actually this means. The definition is that these represent consistent differences among individuals in behavior. That I think it's a pretty tricky definition. So like I will try with a cartoon to explain it. So these friends, Bob and Paul, will then join us for the rest of the journey through the talk. So let's imagine we have two individuals and they're different, as we can see here. One is more aggressive and one is less aggressive. So if we measure them repeatedly and we plot their behavior across a gradient that can be time or context, we expect to observe that on average, these two individuals differ. So we will have Bob on the lower part of the behavioral scoring and Bob on the higher one. So this will be the difference between the two. But it's also true that over a gradient, an animal will fluctuate a little bit in his own behavior. So there is a second component of behavioral variation that happens within an individual and is represented by these parentheses here. So this helps to actually visualize what does it mean to consistently differ in behavior between two individuals. So this kind of highly structured behavioral variation has been observed in a range of animal taxa. I think it's we can say with pretty much confidence that is widespread through the entire animal kingdom. And back in time, people often ask, why do we care? I think maybe nowadays the answer is more simple to provide. This variability in behavior has huge implications on several ecological and evolutionary processes. For example, individuals that are more aggressive and more risk-prone than others most likely are better in competing with rivals and secure mating. Therefore, they might have higher chances to pass their genes to the next generation. But at the same time, individuals that are willing to take more risks in exploring spaces that are potentially dangerous, if they survive, they might actually secure more resources and therefore growing faster than others, and even have more energy to dedicate to other phenotypic traits, as for example, improve their coloration, that we know in many species in the animal realm is an important trait for males to attract females. So, you know, these differences in behavior are linked to many other phenotypes that have an importance in the whole sphere of ecology and evolution. And when I digged into the literature to understand, you know, what was the state of the art, it became pretty clear that a cornerstone of the field of animal personality or behavioral individuality was that this variability between individuals was caused by one or more of these three elements: genetic differences, parental effect, so differences from the experience of the parents that are passed through the offspring via non-genetic bridges, or via environmental factors. And the more I was reading, the more I thought that it was interesting that actually the null hypothesis was untested. So we actually didn't know whether personality or individuality was present in absence of these three elements considered to be as important. And at the same time, this variability was often tested within very small periods of time. And we didn't really have a clear understanding of whether there were developmental effects through which this individuality could change over time. And so I was lucky that just when I was studying the PhD, an important paper was published in which all the populations of Europe of mosquito fish, so Gambusial brucai, that has been my model during the PhD, they've been mapped for their genomics. And they ended up pointing at this population in southern Italy, in Puglia and Torre Castiglione, as the one with the lower heterozygosis. So these individuals were genetically very, very similar to one another. So I went there, I collected these animals, and I carried back to the lab about 200 wild adults. I bred them in the lab in a way that I could use the offspring and standardize basically the experience of their parents. And when I ended up with this offspring, I housed them in very controlled conditions since birth, with the aim of eliminating as much as possible any environmental influence or any effect or experience that would trigger, you know, some trajectory in one individual over another. So basically, these animals were genetically similar. I control for their parental effect, and I try to standardize their housing through their lifetime. And so what I've done is to then measure two of the most classic behavioral axes in the personality literature, that is activity and risk-taking, throughout the lifetime of these animals. And so according with the prediction, I would have expected that in absence of these three triggers of variation, individuals should differ from one another, basically like for small amounts, almost zero. There should be no variance, you know, in absence of genetic, environmental, and paternal effect. But if, for whatever reason, maybe for small genetic differences, some variability did exist, the definition expected this variability to be consistent, so to observe the same differences from birth to the adulthood. And instead, what we found is that we found very low differences early in life, actually no differences at the beginning, but slowly over time, these variations became stronger and stronger up until during sexual maturity, they were like so clearly evident. And by partitioning the variance between and within individuals, we tried to understand what was the mechanism that caused this. And that was interesting because we realized that the differences between or among individuals did not change. So if you look at the thick lines, the blue and the red, these are our two friends, Bob and Paul, they stayed the same. But at the very beginning, these small differences were blurred by a lot of noise around that was represented by the variability within individuals. So individuals were not consistent. They were very chaotic in the way they were doing things. They were collecting information. They were trying to figure out the best strategy to survive in the given environment. And over time, the strategy became more clear. So their variability shrinked at a point in which the difference between the animals became pretty clear. And this was interesting for me, going back to the definition where personality or repeatability is actually a proportion of the variance between or among and within individuals. And so if the denominator goes down, the within individual variance, then necessarily repeatability goes up. So the nice result suggested that variation at the individual level is pretty much inevitable, even in absence of these three triggers, and that actually is not consistent over the lifetime, but rises over development. That were two things that helped me then to inspire like the next studies. So with some colleagues, Clelia that is in the audience, we then thought, is it possible that this mechanism, this kind of a developmental process goes beyond the classic axis of personality variation, so activity and risk-taking, and can be applied to more general behavioral traits, for example, traits related to reproduction. And so we looked at guppies that are known to vary in terms of their reproductive strategies. So females like males that are well-colored, and so certain males will display their beauty, hoping to get accepted by the females and mate. But certain others that do not have that luck will still try to mate by sneaking, and therefore trying to insert some semen into the female. Although it's a more tricky process, it seems to be the best chance that these small males have. So they do differ at the individual level. And we were wondering whether there was an experience component in this choice. So how did the males knew? What was their market value? Which was the best strategy for them to mate? So what we did was actually to expose some individuals to social cues, so allow them to interact with females in presence of competitors to understand how beautiful they were, and therefore which was the best strategies for them. And others, we prevented them from having this kind of social informations. And then we measured the proportion of mating attempt in which they were trying to sneak, so the gonopodium thrust, this is the acronym you will see on the y-axis, versus the actual display. And so what we found with this thick line and the triangles as medians, we found that at the trial 1 in the first week, so before giving any social cues to these animals, the within individual variance, so the plasticity was very high. So males were trying every possible strategy they could in the attempt to mate with a female. But after the first week, when we provided social cues to these individuals, they quickly learned which was the best way for each of them to actually mate. And so we observed this drop in behavioral plasticity within individual variance. Therefore, animals became much more consistent. They stick with a strategy that was more efficient for them in the context. In the non-social treatment, this didn't happen. So again, with the triangles, you can see that the plasticity, the within individual variance stayed pretty much high throughout the entire experiment. So without actual learning what was the best strategy, males kept doing everything they could. So again, this allowed us to understand that there is a developmental process with which individuality emerges, not only in terms of activity and risk-taking, but even in the reproductive strategies of males. So life experience plays a big part. But this, of course, then put at the, arise the question that is then the case that the young animals are so inconsistent in their behavior that they are very vulnerable to variation in laboratory contexts? And so I tried to test this by measuring juveniles, subadults, and adults mosquito fish in 3 different scenarios in which the actual experimental arena simply varied in size. That is something that happened very commonly in the literature. So people measure the same behavior in very different ways. And then I tested each of these animals multiple times in each of these arenas. And as expected, what I found is that it's true. While adults that were more bold, that were more active, were consistently bolder and more active throughout all of the ranges of size of the arena, for the juveniles was not the case. So observing like Bob as the risk-averse individual in one scenario didn't predict Bob's behavior in the next scenario. Actually, things could easily switch. So this basically told us that juveniles are a pretty tricky life stage to look at when you're focusing on these kind of consistent behavioral differences. Then at that time, I think today the situation is a bit different, but a few years ago, a common question was how much this effect that we observed in the lab did actually reflect the variability that was present in the field. And so we looked at this in a lake in the northern Germany, nearby Berlin, where my research institute was at the time for my PhD, by actually capturing these top predators. So these are northern pikes. And we developed an experimental setup to test their behavior in the lab. So actually with the experimental arena nearby the lake. But then we applied ultrasonic tags into the body of these animals. We released them back in the wild, and we measured their behavior throughout like several weeks and months to then match the behavior we observed in the lab with the actual behavior that these individuals showed once they were back in the wild. And we did find, in fact, a strong correlation. So the daily activity rates in the wild for each individuals were negatively correlated with the traits we observed in the lab, saying that the difference between 2 individuals, as strong as we found them in the lab, were pretty well reflected in the difference that we would have found if we would have measured that animals in the wild. That was a good thing to know, we are doing something that potentially has a meaning. But this also has a meaning because, as I inferred a little bit earlier on, with the link between with reproductive tactics and behavior, this variation in behavior is linked to variation in other traits. So this beautiful theory was formulated a few years ago. It's called pace of life, and it's just like an expansion of the classic life history theory, in which behavior and physiology are included into the framework, and they are just applied at the individual level. So basically, Bob and Paul now are at the two sides of this slow and fast continuum, in which behavior or a slow behavior is linked with slow traits that are linked to behavior. For example, Bob most likely will be less active and less prone to take risk than Paul, likely because his metabolic machinery is lower, so does not fuel so much activity as Paul can. This also will occur in a slower growth rate, in a slower reproduction, but by exposing itself to less risk, Bob most likely will live longer. So there is this trade-off between behavior, life history, physiology, that will then allow basically both phenotypes to coexist and to stay over time. Because the classic the classic paradigm is the live fast, die young, that says on one side, if you want to apply it to humans, people that like rock stars or like actors, they become very famous early in life, earn a lot of money, they have a lot of kids, but they might take a lot of risk and incur often in an early death. And if you want to put this into a spectrum, we can find this strategy opposed to us, so the scientists, that instead like might take much longer to secure an economical independence, but we hope that we might have a longer life and more balance. So I think this was an interesting framework, and we try to look at this by asking ourselves whether not only behavior was linked to actual reproductive output, so more offspring, but whether there was even a more intrinsic link of whether the propensity to take more risk was in some way linked to fertility. Since taking risk most likely will incur in an early death, then it would make sense that animals that take risks are more fertile, so that they might produce more offspring quickly, so that in the long run, both the risk-prone and the risk-averse will still survive. And so we did this by looking at sperm traits in male guppies, looking at sperm number and sperm velocities that are commonly proxies for post-mating success, and then looking at risk-taking behavior. So again, is the propensity to take risk in exploring an open space that is potentially dangerous. And we found indeed a weak but positive correlation between the propensity of individuals to take risks and their number of sperms. So males that had more sperms were the ones that took more risks. But again, the question then is this always the case? In the literature, there were plenty of examples in which people tested this kind of assumption, and they did not find such a clear path. And in two of these examples, I actually observed the same. So I've tested pace of life both in the laboratory by using zebrafish and in the wild using pikes, northern pikes. So looking at the behavior in an experimental arena, then measuring the individual metabolic rate as a proxy of their physiology state, and then looking at life history traits related to growth, body condition. And while this diversity in behavior, so this behavioral individuality was present every time, the link between these behavioral differences and differences in life history was not always there. So we found it in the lab, but not in the wild. And the link between behavioral variation and physiological variation was actually never there. So we never found it. So as the literature, we also saw that these assumptions from the pace of life were not always met. And so then the question became, is it possible that the selective pressures experienced in the wild do actually shape this kind of relationship that might actually appear in some contexts, but not in others? And so to test this with the mosquito fish, this time I compared the two populations of mosquito fish that came from very different environments. So the one on the right came from a sinkhole in which the environment was very constant throughout the year, very minimum temperature fluctuation, barely no predators. So the environment was pretty stable. On the right side, instead, I identified a population in nearby Rome that instead suffered from a very strong disturbance, where more than 80% of the adults were harvested every year, and where the temperature fluctuated even up to 20 degrees between the winter and the summer. So then the point here was these two groups of animals, these two populations of animals developed in an environment that were very different. And so my point was, would we find the correlation between diversity in behavior, in life history, and in physiology equally between these two? And to do this, I used the same framework I showed you earlier on. So I captured wild animals, I brought them in the lab, I bred them to standardize the parental experience, and then I used the offspring, put them in a very controlled setup where I could control the life experience from the very beginning of their life until their natural death. And I looked at potential correlations between these phenotypic traits. And in the population coming from stable conditions, the predictions were barely, more or less met, I would say. So we found that individuals that were bolder were also more active than others. They also reached maturity at a larger size, because most likely they were simply eating more, they were just more efficient in conquering resources, and they had a lower standard metabolic rate, so they have lower physiology rates compared to their peers. So there was this pretty structured link between the diversity that individuals had in their behavior, in their life history, in their physiology. On the opposite, in the disturbed population, so the one that was harvested and that experienced the temperature fluctuation, most of these correlations were actually non-existent. And the only one that we found was between activity and size, but was the opposite sign. So in this case, animals that were more active reached maturity at a smaller size. And this made me think of why this could be the case. And then, like, looking back at the environment where the animal came from, things started to become a bit more clear. So if animals respond adaptively to environmental pressures, then when environmental pressures do not support the correlation between these two traits, the correlation should actually not be there. So in this population here, the harvest of the animals purely depended by the size. When they were big, they were captured. There was no rule of boldness. So being risk prone or risk averse made no sense. So at that point, animals simply evolved in a way that they, when they were big, they became much less active, because being active would expose themselves through the risk of being captured, being more visible, or at least this was our interpretation. But this just put the seed in our head that, you know, certain phenomena might not be as universal as we might have thought originally, but they actually might depend by the selective pressures that potentially can shape even this kind of correlations. And then the point was, of course, these two populations belonged to very different environments, so any kind of effect that we have observed arised from evolutionary processes. But is it actually possible that the selective pressure might act much more quickly, even during development? So I've done like a similar experiment where this time I used offspring from the very same population. And again, I started the experiment when they were born. But simply some of these animals were exposed to a low predator treatment throughout their lifetime. So where they were exposed to small amount of fear from their natural predator, that is this largemouth bass, that is the main predator of mosquito fish in the environment where mosquito fish belongs. And then on the opposite side, I developed like another treatment in which this predator treatment was much stronger. So animals were exposed every day to very like strong chemical and visual cues from their predators. And so then what we found, I mean, the paper is still like in preparation, but the results are pretty clear. And what we found is that the correlations between life history traits and behavior were very strong in the first case, where the predator pressure was low. In particular, the correlation between body condition and behavior, so activity from growth rate and activity, from growth rate and body condition, meaning that animals that were in a good state would not move much. Animals that grew a lot would not move much. If they moved much, they also were in a better state. And when they grew faster, so when they invested a lot in growth, most likely they incur into an early death. That is a classic trade-off of assigning energy to competing functions. So either grow fast or developing a strong immune system. But the interesting thing is that all of this correlation were not present in the same individuals. So these are animals genetically very similar, but animals that were exposed to much stronger predator pressures. So you can see here in dark, like the non-overlapping credible intervals mean that the relationship is significant. If they overlap with 0, it's not. And here was never. So we actually found that the pace of life could actually be observed or not in the same group of animals, evolved under the same ecological pressures, but simply living different life experiences. And this was like an important piece of information to then flip the question and say, well, if stress and fear are so important to then shape not only the behavior, but potentially like an array of traits related to behavior, can we use it? Can we actually trigger some reaction as we would like to control species that instead are a problem in the environment? And since mosquito fish is one of the, I think is number 35 of the 100 world worst invasive species, can we use this information to potentially like figure out how we could control them better in the future? So they are, despite their small size, you might all know that they actually are a big problem for amphibians. So they bite the tails and the legs of tadpoles, and the tadpoles basically die. And so in places like Australia, one of the main reasons for the disappearance of amphibian population is actually the colonization of mosquito fish of these water bodies. And so we developed a robotic predator by copying the morphology, copying the locomotion patterns of the buses. And we also equipped the robot with a computer vision system that allowed the robot to understand the world, figure out where the mosquito fish was, and respond in real time. And so we've done an experiment in which we've put mosquito fish and tadpoles together in an arena, and we were testing their behavior, how they interacted with one another, both in presence of a robot and in absence of the robot. The robot here acted as a sort of a bodyguard. So it could discriminate in real time between mosquito fish and tadpoles, and every time the mosquito fish was getting close to the tadpoles, basically interrupt this dynamic by pretending to attack the mosquito fish. As you can imagine, the robot imitating a predator that was a selective predator, actually attacking only the pest, was very effective in controlling the behavior in the short term of the mosquito fish. But what we found also very cool is that weeks after the exposure to the robot, that by the way happened only for a few minutes a week, but a week after this exposure, the behavior of the mosquito fish stayed altered. So in the dark, you see the animals that were from the control treatment, and in the light, animals exposed to the robot. And so the freezing increased, and even the feeding rates increased in a way that we could see the impact of this stress and fear, even like weeks after the exposure, and that had repercussions on other traits, as for example, body condition. Both males and females, after just a few weeks of exposure, lost energy reserves that are typically useful to survive in a challenging environment or to fuel reproduction. And males showed a drop in the number of sperms they had, and females in the weight of their eggs. So this precise action through a robot, so this injection of fear, not only changed the behavior in the short term in the arena with the benefit of the tadpoles, but had a carryover effect in the long run on the behavior and on several life history and reproductive traits of the mosquito fish, showing that robots in the future might not control the pest. This was not the goal of the study, but that using an artifact, as in this case a robot, to simply like trigger a stressful response, allow us to basically unbalance the investment that individuals naturally make between survival and reproduction, where they think that they are attacked by someone that in the reality is not a real predator. They will invest more in survival at the cost of reproduction. And who knows, maybe in the future, this might be inspiring for us to rethink about control practices on species in which, you know, their ecological success seems to be so high that so far we didn't find a solution. But just like to wrap up, just let's keep in mind that playing with the dynamics of ecological system is not always easy, and that does not always turn in our favor. So recent collaboration I had was actually to look at what are the effects of pollutants that, of course, are produced by humans on this diversity in phenotypes that we observed in animals in the wild. And so I learned that some of these pollutants, like the antidepressant Prozac that are prescribed all over the world, are only partially absorbed by our body. And so after the ingestion, most of this product actually get expelled through urine, and our wastewater treatments are very efficient in dealing with the particles that are in the water, but not efficient at all in dealing with these chemicals that are in the water. So they actually make their way out into the environment, and they reach concentrations that are pretty strong. And then the important thing is that our nervous system is pretty well maintained throughout the whole range of animals in the vertebrate groups. And so you can expect that the effect that these medicines might have on us might be very similar to the effect that they might have on the wildlife. And so we tested actually what happened to these fishes that live in the proximity of this area where these chemicals are released. And we look at it by testing these animals at concentrations of these pollutants that are very low. So we're talking about nanogram per liter, so like less than a drop, that are exactly the concentration that have been measured throughout the world, nearby most of the biggest cities. And so here my colleagues have this very nice system in which they have established mesocosms, so with guppies that have been exposed for many generations to either fresh water or low and high concentrations of these pollutants, mirroring the concentration that we actually find in the wild. And so what we did was to then look at these animals and behaviorally phenotype them to try to understand what are the evolutionary consequences of living in a world that is polluted. And so might not be a surprise. What we found is that from the unexposed to the exposed species, we found a drop in behavioral diversity, in repeatability. So animals are very different from one another in fresh waters, but they become very similar when they are in polluted water. And this is pretty easy to explain because this medicine, the Prozac, is designed to cut the extremes. So it basically medicates very extreme emotional states to control this potentially dangerous mental state and put people into sort of a safe average. It does basically the same on fishes. And then when we partition the variance between and within individual, we found in fact that the effect happens at the between individual level. So individuals in fresh water have a chance to differ from one another, but when they live under the effect of this medicine, they tend to behave very similar to one another. So there is this very strong drop in behavioral differences. And for some of you that might remember this old video game called Lemmings, in which basically you had to drive the strategy for your group of lemmings to survive, you would also remember that these individuals could not change the strategy from one another. So if your choice to move the group was wrong, they would all die inevitably one after the other. And so in a larger perspective, reducing the diversity in the behavior of animals is very dangerous because it does not allow them to potentially cope well with changes and diversify their surviving strategies. So just to give an overview of this already pretty long talk, we've been looking at, I've been talking about the behavioral individuality and how this variation among individuals seems to be actually inevitable, not depending only from the classic 3 triggers that have been considered earlier on, but actually potentially there could be a 4th element that is simply caused by development. As a consequence, juveniles are so flexible in the way they behave that they might be very vulnerable to potential artifacts due to lab contexts, and that behavior in the lab, at least, is pretty reliable in terms of explaining the variation that we would observe in the field. Then we've also looked at this paradigm, live fast, die young, that might not always be true. And so it's good to actually understand the evolutionary, but even the developmental pathways of your group of animals to understand whether pace of life should actually be there or not. but all of this, tell us that we can potentially understand better how stress and fear compromise the ecological success of animals, and who knows, maybe this in the future might help us to even reconsider some strategies for controlling animal or others organisms that are pest or anyway problematics. But keeping in mind that the human intervention on nature has severe consequences, and the effect of these global psychoactive pollutants seems to be really bad in terms of competing with the ability of animals to respond to changes. And so, yeah, we should be careful and just keep in mind that diversity is important and we want to keep it. So, yeah. Thank you for the attention, and I just want to thank the two institutions that support me, so both University of Tuscia and the Monash University in Bob's lab, and I'm happy to take any question if you guys have some.
Thank you very much, Giovanni, for this